Hello everyone, my name is Asma from CBO Center for Computational Biology in Mines Paris Tech, and the topic of my presentation today is Multitask Group Lasso for Genome-Wide Association Studies in admixed population. And this is a joint work with Chloe Agathe Azencott. The basic idea of GWAS approach is to look approximately at million positions in the human genome called single nucleotide polymorphisms, SNPs, where different people carry different versions of the genotype. Here, we consider only biallelic SNPs, and our purpose is to compare the genotype of a group of healthy people and another group of people with a disease. The places where the frequencies between cases and controls are different are therefore associated with the phenotype that could be quantitative or qualitative. So GWAS data are presented in microarrays of SNPs with a huge number of features compared to the number of participants included as samples. Many challenges lay ahead in GWAS analysis. First, single marker analysis of classical methods, then population stratification when different ancestry samples are included in the studies, linkage disequilibrium, which is presented by correlation between the SNPs, the computational limitation of complex methods that I will present after, and the lack of stability. So let's start first with single marker analysis. Classic GWAS technique is based on single marker analysis, where each SNP is fitted by a single predictor to obtain a p-value from a statistical test. So this is why there is a need to explore multivariate approaches like machine learning techniques, more precisely feature selections that are based on regularization algorithms. So the regularization term is presented by gamma beta here. We have, for example, Lasso that corresponds to L1 norm regularizations that shrinks the less important features to zero. Then the group Lasso based on L1, L2 norm regularizations, and it is designed to select features that belong to the union of some predefined groups. Also, we present here the multitask Lasso that is equivalent to learn T different regressions simultaneously. So let's talk now about population stratification. That is another problem in GWAS, and it happens in admixed population studies. The problem of population stratification is that the association could be found due to the population structure and not in a disease-associated SNPs. So the state of the art gives us different adjustment possibilities, such as PCA-based methods by including the top principal components in regression models, such as logistic regression or multilinear regression like Eigenstruct. Linear mixed models have been used to correct for this issue, like FastLMM. So now these methods give, unfortunately, an uniform adjustment for all SNPs and can cause overcorrection for some causal SNPs for some population. This is why we decided to explore multitask design, where each subpopulation corresponds to an input task, and then we can perform feature selection model for each task simultaneously. Another problem in genome-wide association studies is linkage disequilibrium, and it is described by the correlation between nearby SNPs observed on the same chromosome. So then we can say that the human genome could be clustered into sets of high LD regions that we will call in the stalk LD groups. We used hierarchical clustering to obtain these LD groups for each chromosome in the genome. So the goal here is to perform feature selection on the LD groups level instead of the single SNP level. So it is known that linkage disequilibrium is influenced by the population structure of the samples. This is why we decided to perform hierarchical clustering for each population separately to respect the LD patterns for each ancestry. And then we can combine both LD groups in order to get a shared LD groups for both populations. Let's see this example. So we have here 4 SNPs in the first group of population 1, and here 2 SNPs for population 2. We allocate, therefore, a first group of 2 SNPs to respect the structure of population 2, and a second group of 2 SNPs to follow also the structure of population 1. And we continue the same procedure for all groups. So now, after presenting solution for each GWAS challenge that we presented, single marker analysis, population stratification, and linkage disequilibrium, we will be able to introduce our algorithm, which is called multitask group lasso for admixed population. The purpose of our method is to set for each subpopulation a task as input. We perform feature selection here using group lasso, where groups correspond to the shared LD groups that we just introduced in the last slide, using hierarchical clustering. And our goal is to select LD groups associated with a disease across all tasks, like it is presented in this graph with the LD groups G3 and G6. We aim also to select some specific subpopulation associated groups, like, for example, G1, G2, G4, or G5. So the problem of sparsity enforcing penalties in general, especially in this complex design, that there are always computational limitations, like memory errors. For this reason, we decided to incorporate safe screening rules that allows the elimination of some features in the optimization in order to speed up the solvers and avoid these memory errors. So the purpose of this method is to estimate the vector of parameter beta by solving the following optimization problem using some geometric properties of the dual formulation. So let's take the example of group lasso. The fitting term is f beta, which is equal to the loss function divided by 2, and the norm alpha beta is equal to the sum of g in capital G of omega g, the norm of beta g. And here, the dual norm of psi is the max of this quantity. Another problem with the feature selection models that they are known to lack stability. We decided to incorporate stability selection technique to improve this index, the stability. So it is based basically on subsampling on random bootstraps with feature selection. The results of all repetition are then aggregated, and this helps to reduce false positives. So the procedure of stability selection is presented by the following steps. We first perform feature selection on random halves of samples. We compute then the stability path, which is the probability of a selection of a given feature k. Here, this probability is denoted by pi k lambda, and it is equal to the probability of k to be in the selected features by the subsample I. Finally, we choose a probability cutoff between 0.5 and 1 in order to keep only the features selected consistently in the set of stable features. Now, I will present some details about the implementation of multitask group Lasso. We used two datasets in order to conduct our analysis. The first one is simulated, including two subpopulations, European and African. So we have cases and controls, and we predefine the disease loci in different chromosomes. For example, here we choose to locate the disease loci in chromosome 2, 12, 19, 21, and 22. Also, we used the real data-driven breast cancer oncoarray data that contains 6 different subpopulations within samples, cases and controls. And to start doing GWAS, we performed some quality control pre-processing steps in order to remove poorly performing SNPs and also to impute the missing genotype values. Also, we used principal component analysis in order to explore the population structure of the dataset and decide the number of the exact number of subpopulation to assign as tasks in the multitask framework. In order to evaluate our algorithm, we used simulation with predefined disease loci. So we will be able now to detect false positive discoveries. We compute also the stability of the selection to evaluate the algorithm. We compared our algorithm to the different scenarios based on the state of the art. So the first scenario is Lasso after PCA adjustment for population stratification at the SNP level. The second scenario is group Lasso after PCA adjustment for population stratification at the LD groups level. Then we explore also separate algorithms such as separate group Lasso for each population at the LD groups level and a separate Lasso for each subpopulation at the SNP level. We also explored the computing time for each method. Now I am going to present some results. Let's start with the simulated data. Here we can see clearly that the multitask group Lasso has the best performance comparing to the other scenarios in the ROG plot. In terms of computing time, we can see on the right that multitask group Lasso is slow comparing to the other methods or scenarios. But this is still reasonable as its framework contains many steps and many components such as hierarchical clustering, the group choice, stability selection. In terms of stability, the multitask group Lasso is more stable than the other methods, with 5,623 selected groups. On the right, also, we can see the effect of subsampling in increasing the stability. So here, another important point is that we can see clearly that the selection on the LD groups level reduce the curse of dimensionality effect as we have less selection choices comparing to the selection on the SNP level. We also obtained similar result with the multitask group Lasso applied to real data. It outperforms the other methods in terms of stability with 62 selected groups. So in this slide, we are presenting a comparison between the Manhattan plot of classic GWAS and the multitask group Lasso selected groups presented by the black dots. So here, each dot corresponds to one SNP per group. We are investigating currently these results from a biological aspect. To summarize, I presented in this talk a multitask group Lasso framework that is based on multivariate approach to consider the effects of SNPs jointly. We assigned also a subpopulation to tasks to address the problem of stratification issue. We used clustering based on LD patterns to deal with the high correlation between the features and to perform the feature selection on the LD groups level using group Lasso. Group Lasso helps alleviate the curse of dimensionality effect in order to have more speed up to our algorithm. We use gap safe screening rules, and we finally incorporate stability selection to improve the stability of the method. So our method is implemented in Python, and the code will be online in GitHub soon. So I'm really very grateful to CBO team, and more precisely the GWAS group of CBO, Unite 900 of Institute Curie, and INR to support this work. So yeah, if you are interested in this kind of research by combining GWAS and machine learning techniques, we have postdoc opening position at CBO, and you can contact my co-author Chloe Agathe-Azencott about it. Thank you for your attention, and I look forward to your question. And also, I am going to present a poster in the ISMB poster sessions of this afternoon, and we can discuss more about my presentation and the topic if you are interested. Thanks.